Welcome to our next video, and our topic for today is mitosis and meiosis. First of all, let us give a brief overview of what is mitosis and what is meiosis. So in mitosis, it starts with cells which has diploid number of chromosomes, and for humans it is 46 in number. 23 from father and 23 from mother, and the result will be two daughter cells with same number of chromosomes, and it is in somatic cells. So, and for meiosis, it has two phases, meiosis 1 and meiosis 2 and it also starts with diploid phase, but when it goes in meiosis 1, it leads to formation of two daughter cells of half the number of chromosomes, that is haploid. This is meiosis 1, and when this goes under meiosis 2, it is equal to mitosis, so it will give the same number of chromosomes, and it occurs in germ cells. First stage of mitosis is interphase. In this, there is a cell with DNA in it, and there is centrosome, which is needed for division in mitosis and meiosis. Then it goes to G1 phase. In G1 phase, it grows bigger by taking the nutrients from the environment. This is G1 phase. Then it goes to S phase, and S stands for synthesis phase. Here the main event that occurs is the duplication of chromosome and also the duplication of centrosome. And this is centrosome. So this is S phase. Then it goes to G2 phase. In this, the cell grows even bigger, and all the other organelles also duplicate in this cell. Remaining the chromosomes are duplicate, and centrosomes are same as S phase. So this is G2 phase. After interphase, our next stage is prophase. In prophase, there is condensation of chromosomes, and the nuclear membrane starts to disappear, and the centromere here starts to move to the poles. So here. So the general points are condensation of chromosomes, nuclear membrane starts to disappear, centrosomes move to opposite poles. Our next phase is metaphase. In this stage, chromosomes are highly visualized because they are in the most condensed form. So in this stage, the sister chromatids line up, and it is called equatorial plate, and these are centrosomes. They extend the microtubules and form a mitotic spindle. This is the equatorial plate formation. So the general points of metaphase is nuclear envelope is completely gone. Sister chromatids line up and they form an equatorial plate, which is important for this stage, and formation of mitotic spindle starts. Next stage is anaphase. In this stage, sister chromatids are pulled by the centrosomes to their sides, like this one. This is mitotic spindle, this is breaking. So, and the point of attachment here, this is called kinetochore. So the general points here of anaphase is sister chromatids pulled apart by the mitotic spindle. That is, one of the chromatid goes to one side and the other chromatid goes to another side. And other chromatid goes to other side. Next phase after anaphase is telophase. In telophase, at each pole, full set of chromosomes gather, like this. And there is the formation of nuclear membrane, that is, nuclear membrane starts to appear again. So the general points here are chromosomes gathers at two poles, and nuclear membrane starts to appear. After this, the two cells need to pinch off. So this stage of pinching off is called cytokinesis. After pinching, they form daughter cells with two pairs of chromosomes. So this is all for mitosis, and now we will discuss about meiosis. Discussing about meiosis, first of all, it has two phases: meiosis 1 and meiosis 2. Meiosis 1 is also called reductional division, because it divides the diploid number of chromosomes into haploid number of chromosomes. Second is just like mitosis, and it is called equatorial division. After this, they have same number of chromosomes. The daughter cells have same number of chromosomes, so it is called equatorial division. First phase is interphase, and interphase is just like mitosis. It has three stages. G1 phase, in which the cell grows bigger by the help of nutrients from the environment. Second stage is S phase, which is synthesis phase, and the main event occurring in this phase is duplication or replication of chromosome. Then the third phase is G2 phase, where all the other organelles duplicate. Second phase after interphase is prophase. This is different from mitosis because it has further stages, like first one is leptotene, second zygotene, third pachytene, fourth diplotene, and fifth one is dikinesis. And then the next phase after prophase is metaphase. So here we are discussing about meiosis 1. So this will be called prophase 1 and metaphase 1. Next stage is anaphase 1. After anaphase comes telophase 1. We will start discussing from the prophase 1. So starting from prophase 1 stage of meiosis 1. First is starting from prophase 1, which is a stage of meiosis 1. First step in prophase 1 is leptotene, or it is also called leptonema. Here lepto means thin and nema means threads. So from here we can conclude that the chromosomes appear in form of thin thread, and it can happen only when there is condensation of chromosomes. In second stage of prophase 1, it is zygotene. Zygo means pairing. So here in a cell, two sister chromatids separate and then synapsis occurs. What is synapsis? Synapsis is pairing of two sister chromatids, like this shown in the figure. Third stage is pachytene. Here is the formation of tetrad. That is, 4 chromosomes are together, and here the center point is called centromere. So here the general points are formation of tetrad, that is 4 chromosomes together, and then sister chromatids overlap each other in this phase. In this phase, after the stage occurs crossing over. It means some parts of the maternal chromosome gets attached to some parts of paternal chromosome here, and this point of attachment is called chiasma. So the general points are: crossing over occurs, so there is mixing of maternal and paternal chromosomes takes place in this stage. Next stage of prophase 1 is, which is the fourth stage, it is diplotin. In this, there is first of all partial disappearance of the nuclear membrane, and the chiasma starts shifting towards one end, and this is called terminalization. So the general points are: partial disappearance of nuclear membrane or nuclear envelope, and chiasma starts shifting towards one end. And this process is called terminalization. Next stage after diplotin is dikinesis. In dikinesis, genetic recombination took place. Like, these are separate, but after crossing over, some part of maternal chromosome comes over paternal chromosome, and some part of maternal chromosome comes over paternal chromosome. So the general points are: genetic recombination, and mitotic spindle starts to form here, starts to extend. So this is all for prophase 1. Our next stage is metaphase 1. Here the chromosomes line at the center, and the centrosome are at the opposite poles with their mitotic spindle. So the general points here are: chromosomes line up at center, and again it is an important event which is called formation of equatorial plate, and centrosomes start forming mitotic spindle. Next phase of meiosis 1 is Anaphase 1. In this phase, this is centrosome, and the pair of chromosome is pulled towards one end by this centrosome, and the other chromosome by the another centrosome. So the general points are: pair of chromosomes are pulled to opposite poles, and here notice that in mitosis the Anaphase stage had chromatids separated. Sister chromatids were separated in mitosis, but in meiosis, full chromosome is shifted to opposite poles. So this is the difference between Anaphase stage of mitosis and meiosis. And the next stage of meiosis 1 is telophase. Telophase 1. In this phase, chromosomes move towards the opposite ends of the cell, and the nuclear membrane starts to appear again. So the general points here are: chromosomes completely move to opposite poles, and nuclear envelope starts to appear. After telophase 1, again like mitosis, the pinching off of this cell will start to occur, and this is called cytokinesis. This will give rise to two daughter cells containing half the number of chromosomes. Now we discuss about meiosis 2. As I said earlier, it is same as mitosis, and it has the same stages or phases like prophase. This will be called prophase 2 because of meiosis 2, metaphase 2, anaphase 2, and then telophase 2. After telophase, there will be cytokinesis again. This time, one haploid daughter cell that was the result of meiosis 1 will give rise to two daughter cells. So there will be formation of 4 haploid cells as a result of meiosis. These 4 cells, in males, they form sperm cells, and in females, one form the egg cell and other 3 form the polar bodies. This is all about meiosis.